Okay, so here we are in chapter 17. We're talking about gene expression, and we are going to talk about how we go from that DNA that we just talked about in chapter 16 to making the protein. So if you remember all the way back from chapter 6, we learned that DNA contains the instructions for making proteins. And so in this chapter, chapter 17, we're really going to talk about how we do that. What are the processes that we go through to start from DNA to get to protein? All right, so the first thing we need to do is kind of get the overall big picture in our mind. And in biology, we refer to this process as a central dogma. It is a well-believed idea of how things work, and everybody sort of accepts this, and it's just a well-understood process. So we, last time in chapter 16, kind of left off with DNA. We talked about replication and the way that DNA is copied during cell division. And so we are going to start here in chapter 17 and work our way through to proteins. So again, remember back from chapter 6 when we did the tour of the cell, we said that DNA contained the instructions for making our proteins. So if you guys remember back to the analogies that we did back in chapter 6, DNA was our recipe, or what we used to make our widgets or whatever it was that we were making. Okay, so the way we get from DNA to proteins, the first step is we go through a process called transcription. And so we're going to talk about that in this chapter. And this is sort of an intermediate step where we make RNA. So we transcribe or make a copy of that DNA to produce an RNA molecule. The second step that we'll talk about is called translation. By the way, let me back up. Transcription happens in the nucleus. So all of transcription takes place in the nucleus because that's where the DNA is. The second step that we talk about is called translation. Translation takes place in the cytoplasm, specifically at the ribosomes, because remember, the ribosomes are where we make the proteins. Remember that all the way back from chapter 6. The proteins are made in the ribosomes, and so translation takes place in the ribosomes because that is the translating of the translation process converts RNA into proteins, and so that is when we actually make our proteins. So remember, proteins can be structural, they can be enzymes, and proteins are what produce the phenotypes that we've been talking about in chapters 14 and 15. So that sort of ties together a whole lot of chapters that we've been talking about. This one slide sort of ties together a whole lot of the chapters that we have talked about up until this point. So we go all the way back to chapter 6, where we talk about DNA being the instructions for making the proteins, and proteins being made in the ribosomes, and then we bring it back to even chapters 14 and 15, where we were talking about our genetic stuff. So it is these proteins that we make from the DNA that produces the phenotype that we see when we talk about genetics. Okay, so let's talk real quickly then about a couple of differences between RNA and DNA. So we talked about this double helical structure of DNA a lot last time in chapter 16. And we said that DNA contained this sugar called deoxyribose. And it was that carbon 2 that lacked the OH group that made that sugar deoxyribose. And we talked about the bases in sugar, or in DNA, being A, T, C, and G. So a couple of differences here then, and we talked that DNA was double-stranded. A couple of differences here then with RNA. RNA contains the sugar ribose. In RNA, an A pairs with uracil instead of thymine. So in RNA, there is no thymine. Thymine is replaced by uracil. And then RNA is most of the time single-stranded. We do know now that there are some double-stranded RNAs, but for the most part, RNA is going to be single-stranded. Okay, so then we also want to talk about a couple of differences between transcription and replication. Because if you remember, I said that when we transcribe DNA into RNA, and if you think about that word, transcribe, it basically means to make a copy. If I asked you to transcribe a letter that I had written, that would be asking you to make a copy. And if we think about replication, that's the same thing. It's making a copy of the DNA. So how are these two processes different? Well, they differ in three main ways. Number one is the amount of DNA involved. When we talked about replication, we were replicating the entire DNA molecule, every single bit of that DNA, because we're getting ready to pass that DNA down to a daughter cell. And so we have to have every single bit of that DNA copied. When we talk about transcription, we are only transcribing certain parts or selected genes within the DNA. The analogy that I like to use for transcription is a cookbook. So let's say that I have my great-great-grandmother's cookbook, and in that cookbook she has a recipe which she has written down for her chocolate chip cookies, and she makes the best chocolate chip cookies there are. If I were going to make those cookies, if I were going to go into the kitchen and make my great-great-grandmother's chocolate chip cookies, I would not want to take that cookbook, that precious cookbook, into the kitchen with me, because I might spill something on it, I might catch it on fire, who knows, it could get damaged. So what I would do is I would make a copy of that recipe. Now, I wouldn't copy the entire cookbook, because I don't need the entire cookbook. I only need the recipe for chocolate chip cookies. And that is transcription. So in replication, yes, we would be making a copy of the entire cookbook. And I might do that if I wanted to pass that cookbook down to my daughter so that she could have those recipes. I maybe would make a copy of the entire cookbook and pass that down to her. But if I'm going into the kitchen to make chocolate chip cookies or proteins, then I only need to copy down the recipe for the chocolate chip cookies. And that is the process of transcription. You only transcribe the pieces of the DNA or the parts of the DNA that code for the proteins that you want to make. Now, the enzymes are also different. So in DNA, in replication, we use DNA polymerase as our enzyme to bring in our complementary nucleotides. In transcription, we use RNA polymerase to do that. And then our end product is also different. We know with the replication, we're going to end up with that double-stranded DNA molecule, whereas with transcription, we're going to get that single-stranded RNA molecule. Okay, so a little bit about the process of transcription. So again, this is converting DNA to RNA, and specifically mRNA, where the M stands for messenger. And so we'll talk about what that means in a second. But the M is for messenger RNA, which actually contains the code for making the proteins. So a couple of things happen. We're going to get an enzyme that's going to come in and unwind a section of the DNA. Now we know what this enzyme is because we talked about it in the last chapter. It is called helicase. So this is the enzyme that comes in and separates our two DNA strands. We then have RNA polymerase that brings the complementary RNA nucleotides to the DNA strand. So if you see here, we sort of have this hybrid a little bit where we have a blue strand, that's our DNA. We have the R strand, that's our RNA. So we sort of get this DNA-RNA hybrid where we have complementary base pairs, and that is catalyzed by the enzyme RNA polymerase. So we actually create, when we carry out transcription, is something called a pre-mRNA that we have to process in order to turn it into a mature mRNA. So we'll talk about what the processing involves. Okay, so in order to process a pre-mRNA, we have to do three things to it. The first thing we have to do is cut out what are called introns. So there are sections of the DNA strand. Some are called exons. Some pieces or sections are called exons, and some are called introns. We know that the parts of the DNA that actually contain the code for the protein, that decode the protein that we want to make, those are the exons. So the exons contain the codes. The introns, we used to think they were all just junk, just useless pieces of DNA. We're now learning that that's not so much the case, that they can have regulatory functions. Some of them are just junk. I'll be honest, some of them are just junk, but a lot of these introns have sort of regulatory functions, and we're sort of learning a little more about these day by day as far as what they are. But in order to translate the RNA and make our protein, we don't need those introns. So we cut them out. So the introns are eventually going to get cut out when we make our mature mRNA. Now, two other things have to get added to the ends of this DNA. So we add a cap on the 5' end, and we add what's called a tail on the 3' end. So the cap is what's going to allow our mRNA to bind to the ribosomes. Remember the ribosomes, that's where we make the proteins. And so we know that that mRNA is going to have to interact with that ribosome in order to make the protein. The tail is going to dictate how long that mRNA is going to last inside the cell. So a longer tail, a longer lifespan inside the cell. A shorter tail, a shorter lifespan inside the cell. So three things that happen. We cut out the introns, we add the 5' cap, and we add the 3' tail. Alright, so we now have a mature mRNA, and we're ready to decode this mRNA into protein. And you can see my sequence kind of got messed up a little bit here on this slide, so we'll talk about this in class and I'll work it out for you a little bit better. But what we have is what's called the genetic code, and this is a chart that has been worked out years ago and is widely accepted. And so basically the way that we read the code for the mRNA is 3 letters at a time. So we read the first letter, which is A-U-G. So that's our first word. So each word has 3 letters, and we read this one word at a time, or 3 letters at a time. So there are 64 words in our language here, and they each have 3 letters. So if we figure out how to decode this, you can see at our chart we have first base. We have first base here, we have second base across the top, and then we have third base over here on the right hand side. So if we were to take A-U-G as our first word in our series, we would come over here to A, that's our first base, and then across the top we come to U, and so we know that we were working somewhere in this box right there, and then we would go over here, our third base, to G. And so we know that we are working right here in this box because that's where all 3 of those letters meet. And so that is the start codon, or it codes for the amino acid methionine. So AUG is our only start codon. So for every mRNA transcript, they're going to start out with an AUG. Now if we look at our next word in our language, we have UGA. UGA is our next word. And so if we look at where those come together, we come over here for U. So we know we're somewhere in this block here. G is our second base, so we know we're somewhere in this block for UG, and then our last block is A, UGA. So we come right here, and UGA is one of our stop codons. So there are actually 3 stop codons. You don't have to memorize them, but you do have to know how to use this table. So I would ask you, when you are studying, that you pay attention to and understand how to use this table and how to decode the mRNAs, and we'll talk more about that in class. Alright, so once we get our mRNA transcribed, it's now time to translate it. So remember, translation happens in the cytoplasm at the site of the ribosomes specifically. And there are three stages or phases of translation. We have initiation, elongation, and termination. So we're going to walk through those briefly so that we get a sense of what's going on in each of these phases. Okay, there are a couple of other RNAs that we're going to need to carry out translation. There are the tRNAs, which I like to refer to as the amino acid taxis. And so their job inside the cell is to deliver the amino acids to the site of the growing peptide chain. So they're bringing amino acids to us so that we can make this big long peptide. Amino acids have two really important features to them. Number one is the anticodon. This is what is going to base pair with the codon of the mRNA. So that is that three-letter word, that AUG or GGA or CGU, whatever our letters are that make up our words, that make up our codons. The anticodons base pair with those codons in a complementary way. The other important feature of the tRNA is the amino acid site. So that is where the amino acid binds to the tRNA and how it is moved around in the cell and how it is delivered to us so that we can build our protein. The other type of RNA that we need is the rRNA. So this is, by the way, tRNA is known as transfer RNA, and rRNA is ribosomal RNA. So that is RNA that makes up the ribosome. So we know we're going to need that ribosome in order to make our proteins, and that is made up of RNA. Alright, so let's walk through the phases, or the stages of translation. The first stage being initiation. So we need a couple of things to happen for initiation. Number one, we need the ribosome. So that's our first step. We have to have that ribosome present in order to translate our mRNA. Number two, we need our mRNA. So we need to have transcribed a piece of the DNA. We need our mRNA transcript. And remember, that mRNA transcript is going to start with AUG, or our start codon. The third thing we need is our initiator tRNA. So remember, AUG codes for methionine amino acid, and so that is the first amino acid, that's the first part of our growing chain for every peptide chain. So we need the three of these things to kind of come together and bind and snap together all in one place, and that is known as initiation. The next phase of translation is elongation. The ribosome is going to read the sequence of mRNA codons. So remember, our first one was AUG. Our second one might be CGA. So it's going to read this codon, and it's going to bring the appropriate amino acid to the chain that codes for, that is coded by that codon based on the genetic code. So the amino acids are going to form a bond, and the tRNAs are going to be recycled and released and reused in the cell, and the ribosome is going to translocate down the mRNA to read the next codon. The next one might be ACC. Okay, so here's our next codon, and it's going to code for another amino acid. So that amino acid is going to be brought into place, and we're going to form a bond with that amino acid. So we now have a tripeptide with 3 amino acids bound together. This keeps happening until we reach one of the 3 stop codons. Alright, and I will show you a picture, a little video of this in class, so that this will make more sense to you and you can see the things kind of moving around. It doesn't work so well here in this setting, but I will show that to you in class. The last phase of translation is termination. So remember, this ribosome is going to translocate down this mRNA, reading 3 letters at a time, until it comes to the stop codon at the end of the transcript. That's the end of the mRNA and signals that translation is done. So what happens is, when we reach that stop codon, the ribosome, the mRNA, and the growing peptide, or the peptide chain or the amino acid chain, detached from each other. The ribosome can go and be recycled and reused. So this is going to be reused inside the cell. The mRNA is going to be degraded because it has done its job. We've done what it has done, what we've asked it to do. It's made the protein. And so it's going to be degraded and destroyed inside the cell. And then the amino acid chain is going to become that protein. Whatever that protein is that we were making, we're going to use that protein either inside the cell or ship it outside the cell. And that's going to become our protein. So we have completed the process of translation. Now, we all have heard about mutations in DNA and kind of what that means, and usually we think of them as being very bad, and most of the time they are bad, but we need to know that mutations in DNA can be harmful, they can be neutral, they can have no effect whatsoever, or they can even be beneficial. And so we don't always think of mutations as bad things, but they do alter, usually, the proteins that we make. So for example, if we have a particular DNA sequence that's represented here, we would get a specific mRNA sequence based on the complementary nucleotides that we have in DNA. And based on this DNA sequence, we would get a protein sequence, a primary sequence of our protein. So in this case, we have threonine, proline, glutamate, lysine. That is the primary sequence of our protein based on the sequence of the nucleotides of our DNA. If, for example, we had a mutation, and so in this case, we had a deletion mutation where we lost this T nucleotide. So we now have CCC instead of CTC in our DNA strand. So you can see how that's going to change the sequence of our mRNA. So we now have GGG in our mRNA instead of GAG. And then we have AGA instead of GAG. And then we just have AG down here. So we have an incomplete codon or an incomplete word at the end. Now you see how the first two amino acids are not affected because their mRNA sequences were the same. So we still have threonine and proline. But now instead of glutamate, we have glycine and arginine. So we have not made the protein that we originally intended to make. We made something different, we made something incomplete. It may be completely non-functional, or it may have altered functions than what our original protein was intended to have. So mutations, when we talk about mutations, we are talking about changes in the sequence or the ordering of the DNA bases. And so a change in DNA would have a direct effect on the protein that is produced. Now, mutations can be caused by X-rays, they can be caused by UV light, by free radicals, by chemicals, all sorts of things can cause mutations in the DNA. And remember, these mutations can be harmful, they can be neutral, or they can be beneficial. So that is the end of chapter 17. I will see you in class.